DreamFold, where we develop world models for drug discovery. We have been motivated by the fact that there are still out there billions of people affected by incurable diseases, in particular diabetes, cancer, neurological diseases. This creates a huge loss of money on failed clinical trials and years of research. The problem arises that conventional drug discovery often follows, let's say, obsolete pipelines, and the hits discovered there often fail to translate clinically. The reason is that they focus on static protein structure instead of learning the physics-based, let's say, conformational changes of proteins. So the solution is DreamFold, where we have developed the first world model for drug discovery to drive drug discovery on conformational ensemble. The team is composed by 2 postdocs at Yale and Professor Mark Gerstein. So the pipeline that we propose makes use of these world models, which are the latest breakthrough in AI, as transformers were a few years ago. So it is basically a method to compress complex reality into a latent space, which is often called the dream environment. And we developed the first physics-based world model to do protein simulation, and we have also developed tools that follow to perform virtual screening of millions of drugs, as well as drug optimization, with the hope of unlocking new therapeutic possibilities on otherwise undruggable targets. Our method is basically a lot faster than what is already out there, very accurate, allows drug discovery on alternative targets, and it is a full pipeline that goes from target identification to drug optimization. Our venture strategy will be to do strategic collaborations with pharmaceutical companies, but also develop internal drug discovery pipelines that will allow us to have the IP full ownership of the drugs that we develop. So where we stand right now is that we have thoroughly designed, trained, and validated this world model on a variety of protein targets. We have filed a patent of the model. We have also worked on the additional tools for drug optimization. We are going to file an additional patent on that. So what we are planning to do for our next goal is basically looking for a pre-seed to help us securing some partnerships with pharma, develop one or a few internal drug programs, increase the team size to bridge AI and experimental biology, and set up a team for the experimental validation. For our longer-term goal, it will allow us to create an impact on society and scale up our technology for a variety of diseases. And thank you very much for the attention.
Thank you. AI for drug discovery is a very crowded space. Can you help us understand how your solution's differentiated from the other solutions out there?
There are many tools out there, such as AlphaFold, that everybody knows that work on protein structures, but there are not many tools that are able to, let's say, solve the physical complexity of how proteins fold, how proteins are, because they are dynamic entities. They keep moving. They're not a single static structure like AlphaFold predicts. So we are one of the first that have developed these world models, which are extremely efficient. They are up to 30,000 times faster than other physics-based methods such as molecular dynamics, but they still maintain the structural accuracy that is necessary to drive these structure-based drug discovery studies. And so this is what we are planning to do, have a very accurate but very fast method.
So just to follow up, so it's tied to the fact that this is a world model and this is deterministic as opposed to probabilistic. Is that a simplification?
You can say that, yeah. But also one key aspect is that it needs a very little amount of training data. It doesn't rely on millions and millions of structures to be trained. So you can sort of train your model to purposely address a specific protein target in a very efficient manner.
Great. One last question.
I guess what's the engagement with industry been to date? You talked about biopharma and others as partners. How have you started to explore that in terms of your patented work?
I I think it will be best suited for proteins that people have tried to create key drugs but have failed, for example, neurological diseases. I think there will be a lot of interest to apply our method to study protein that misfold, for example, in the cases of Parkinson's and Alzheimer's. A lot of years have been spent on trying to cure these diseases, but there's a lot of failure rate. In our case, we are able to identify these alternative structures to work on, and I think there will be interest from pharmaceutical companies to have a branch collaborating with us and drive the pipeline together.
And have you kicked off those conversations yet?
Sorry?
Have you started those conversations? Okay.
Well, we have performed internal validations against literature data and experimental and computational data. What we are planning to do in the short term is to work with other laboratories at Yale to do a proper experimental validation to see how well it works.